Hello everyone. So in this video we will discuss the question number 47 of GATE 2011. So the question is asking HAT, which is composed of hypoxanthine, aminopterin and thymidine, is used for selecting the hybridoma based on the following. Statement 1. Only hybridoma will grow since it inherited the AGPIT gene from B cells and can synthesize DNA from hypoxanthine. Statement 2. Myeloma cells will not grow in culture since de novo synthesis is blocked by aminopterin and due to the lack of AGPIT gene. So let's see what is the criteria for the selection of hybridoma cells. So in this case, from this diagram you can see that this mouse was challenged with antigen, and after the antigenic exposure its spleen cell was collected, and from the spleen cell B cell were collected, and it was fused to the myeloma cells, right, which are the tumor cells. So upon the fusing, the hybridoma will be occurred. Hybridoma or the hybrid cells which will be formed by the fusion of the B cell and myeloma cells will be formed. Now there are three possibilities. One, there will be fusion of B cells and myeloma cells which are called as the hybridoma. There might be unfused B cells, there might be unfused myeloma cells, right? So now we need to select only the hybridoma from the unfused myeloma cells and unfused B cells, right? So for selection we will culture it in a HAT medium and we will select for the positive, which are the hybridoma cells. So what is this HAT made for? HAT is composed of hypoxanthine, which is a precursor for the DNA synthesis, aminopterin and thymidine. So this medium contains aminopterin, which inhibits the de novo pathway of nucleotide synthesis. So now if the media has this component aminopterin, so now in this case the cell will be dependent on the salvage pathway for DNTP synthesis, right? So the cells will be dependent for the salvage pathway for nucleotide synthesis, and the salvage pathway requires AGPIT gene. So and this AGPIT gene is so only the B cells it has the AGPIT gene. So the unfused myeloma cells will die because will die in this HAT medium because they lack the AGPIT gene. So as the media contains aminopterin, so in the absence of AGPIT gene it cannot use the salvage pathway, so it will die. Unfused B cells will die although they have the AGPIT gene. So for some generation they will survive in this HAT medium, but after that they will die because B cells are not immortal, right? So only fused cell, which the fused cell or the hybridoma cell which actually inherit the AGPIT gene from the B cell and the immortality from the myeloma cell, right? So only those cells can survive which inherit the AGPIT gene, so that it can produce its nucleotide by the salvage pathway, and it has inherited the immortality feature from the myeloma cell. So now only fused cell will survive in this HAT medium as they have AGPIT gene from B cells and immortal property from the myeloma cell. So if you will see the options, then we can see that only hybridoma cells will grow since it has inherited the AGPIT gene from the B cell and can synthesize DNA from the hypoxanthine, so it is correct. And myeloma cells will not grow because its de novo synthesis is blocked by aminopterin and it does not have the AGPIT gene. So that's why the myeloma cells cannot grow in this HAT medium. So option C, which says that both of the statement 1 and statement 2 are true. So C is the correct option here.